What if I told you 10 discoveries in biology quietly rewired how we eat, heal, and protect the Earth? From vaccines and DNA to climate-smart crops and gene editing, this is the story of ideas that fed billions, saved millions, and might just save our future. If food, science, and the future medicine excite you, hit subscribe and ring the bell. Stick around for number 2 and number 1 because they're still rewriting tomorrow. We start with the engine of life. At number 10, the photosynthesis. Life on Earth is almost impossible without photosynthesis. Scientists uncovered how plants turn light energy by splitting water and absorbing carbon dioxide into sugars, and later mapped the Calvin cycle. We learned how to boost crop yields, breed better crops for different light conditions, and forecast the carbon cycle that governs climate. It improved crop physiology and yield optimization, an inspiration for biosolar materials and oxygen therapy insights, laid a foundation for reforestation and carbon sequestration models. One fact: the total amount of energy captured by photosynthesis on Earth is staggering, about 130 terawatts. This is approximately 6 times more energy than the total power consumed by all of human civilization combined. Next, the nitrogen cycle and biological nitrogen fixation, the reason legumes enrich soil. Discoveries about rhizobium bacteria and mycorrhizae showed how microbes feed plants, reducing fertilizer dependence. Crop rotations with legumes, biofertilizers, healthier soils. Microbial metabolism insights for probiotics and diagnostics. Legumes help in reducing runoff, cleaner rivers, lower nitrous oxide emissions. While humanity's industrial Haber Bosch process consumes 1 to 2% of the world's entire energy supply to produce fertilizer, symbiotic bacteria living in the root nodules of legumes perform the same nitrogen-fixing process at room temperature and pressure, powered only by the plant's sugars. At number 8 is the laws of inheritance discovered by Gregor Mendel without any knowledge of DNA, genes, or chromosomes. His discoveries were a feat of pure abstract reasoning and statistics based on observing patterns in pea plants, and his work was largely ignored by the scientific community for over 30 years. Mendel's peas gave us the rules of inheritance. Later, quantitative genetics turned breeding into a predictive science. Before DNA, this powered hybrid corn, disease-resistant rice, and better dairy herds. Its impact on food and agriculture mean faster, targeted plant and animal improvement. For medicine, risk scoring for inherited diseases, pharmacogenetics groundwork. Impact on environment huge by breeding for drought and heat tolerance to protect ecosystems. At number 7 is the natural selection. It explains pesticide and antibiotic resistance, guides conservation genetics, and tells us why we must rotate crops and therapies. Evolution can occur remarkably fast. When industrial soot darkened trees in 19th century England, the peppered moth population evolved from mostly light-colored to over 98% dark-colored in just 50 years to maintain its camouflage against predators. Natural selection emphasizes on significance of integrated pest management in food and agriculture. For evolutionary medicine, why pathogens adapt and how we counter. It also taught us that preserving genetic diversity to keep species resilient. Up next is the cracking the double helix, and the genetic code turned life into readable information. That leap connects every item on this list: breeding markers, gene tests, vaccines, marker-assisted selection, tracing crop origins, genetic testing, rare disease diagnosis, targeted therapies, DNA barcoding for biodiversity and anti-poaching. Do you know that the amount of DNA packed into a single human cell is immense? If you could uncoil and line up all the DNA from all the cells in your body, the resulting thread would stretch from the Earth to the Sun and back more than 300 times. At number 5 is the PCR in genomics, which made copying DNA trivial. Later, high-throughput sequencing made reading it cheap. Suddenly, we could identify pathogens in hours, track outbreaks, and map the microbial world. Food and agriculture, rapid plant and animal disease diagnostics, traceability, medicine, COVID-era testing, cancer panels, prenatal screens, environment, eDNA to monitor invasive species and endangered wildlife. The polymerase chain reaction, PCR, is a molecular photocopier of incredible power. A single cycle doubles the amount of target DNA, meaning that after just 30 cycles, a few hours, one starting molecule can be amplified into over 1 billion identical copies. Startling fact: PCR can amplify DNA from essentially a single molecule to billions of copies in hours, and the cost to read a human genome has dropped from $3 billion to well under $1,000. Germ theory is placed at number 4, which proved microbes cause disease. That unlocked vaccination, antiseptics and pasteurization, transforming hospitals and our kitchens. Food/agriculture, safer milk and canned foods, protected livestock. Medicine, from smallpox to modern vaccines, public health's backbone. Environment, cleaner water systems, safer waste management. Before Louis Pasteur's germ theory, surgeons took pride in their blood-caked operating coats as a symbol of experience. They had no idea they were transferring deadly microbes between patients. And something as simple as hand-washing reduced surgical mortality rates from over 40% to single digits in some hospitals. Startling fact, smallpox killed an estimated 300 million people in the 20th century, and vaccination drove it to 0 cases worldwide. At number 3, the discovery of antibiotics, which saved tens of millions of lives and protected animals from deadly infections. But evolution bites back. Antimicrobial resistance is rising, demanding stewardship. Food and agriculture, prudent use in livestock, biosecurity, vaccines for herds. Medicine, new drug discovery models and combination therapies. Environment, wastewater monitoring to track resistance genes. The discovery of penicillin was a complete accident. In 1928, Alexander Fleming returned from holiday to find that a mold, Penicillium rubens, had contaminated a petri dish and was visibly dissolving the colonies of Staphylococcus bacteria around it. Startling fact, penicillin resistance was reported by 1943, just a year after widespread use. Today, antimicrobial resistance already kills hundreds of thousands annually and is rising. The Green Revolution is at number 2, which means in the mid-20th century, high-yield, disease-resistant varieties, breeding, and agronomy practices. It prevented mass famine. Modern breeding, speed breeding, and genomic selection carry it forward. Wheat and rice yields soared, now climate-smart traits. Better nutrition, biofortified crops combat hidden hunger. Higher yield per hectare can spare land, if managed well. Green Revolution is often credited with overcoming famine for approximately 1 billion people, as wheat and rice yields doubled to tripled in many regions. The semi-dwarf wheat varieties developed by Norman Borlaug didn't just have higher yields. Their short, sturdy stalks could support heavy heads of grain without collapsing. This simple architectural change was a key innovation that helped double or triple wheat yields, saving an estimated 1 billion people from famine. Finally, at number 1 is CRISPR as gene editing tools. A bacterial immune trick turned gene editor that lets us precisely change DNA. It's enabling bacterial blight-proof cassava, drought-tolerant rice, non-browning produce, gene therapies for inherited disease, and even tools to curb vector-borne illnesses with strong ethical guardrails. Food and agriculture, faster trait development without dragging along unwanted DNA. Medicine, from sickle cell cures to personalized oncology. Environment, potential to rescue endangered species genetics, control invasives. The CRISPR system that has existed for millions of years. Bacteria use it to capture snippets of viral DNA, which they then embed in their own genome as a most wanted gallery to recognize and destroy future invaders. In 2023, the first CRISPR-based therapy won approval for sickle cell disease, and a single CRISPR edit can change one letter out of our approximately 3 billion letter genome with surgical precision. From light harvesting leaves to editing genomes, biology's greatest hits shaped what's on our plates, what's in our pharmacies, and what's possible for the planet. Which discovery deserves a deeper dive? Comment the number, and I'll make a dedicated episode. If you learned something today, like, subscribe, and share this video with a curious friend. Science isn't just in textbooks. It's food we eat, pills we take as a cure, and the air we breathe to freshen up. This was your shortcut to the frontier in modern biology. Thanks for watching the video. If you find this information as incredible as I did, hit that like button and subscribe for more deep dives into the ideas and stories shaping our world. See you next time. Stay curious.